Concept of species. A species is a reproductively isolated population that shares a common gene pool and a common niche. This definition defines a species reproductively, genetically, and ecologically. The definition of a species given above is taken partly from Meyer, is somewhat idealistic. Since it assumes sexual reproduction, it leaves the term undefined for a large class of organisms that reproduce asexually. Biologists frequently do not know whether two morphologically similar groups of organisms are potentially capable of interbreeding. Further, there is considerable variation in the degree to which hybridization may succeed under natural and experimental conditions, or even in the degree to which some organisms use sexual reproduction between individuals to breed. Consequently, several lines of thought in the definition of species exist. 1. In 1. In practice, species are defined by easily recognizable phonetic characters that reliably indicate what species an individual belongs to. 2. The biological species concept defines a species as a set of interbreeding forms. Interbreeding between species once prevented by isolating mechanisms. Principal authors John Ray, Buffon, Dobzhansky 1935, Meyer 1942. Such species are also termed as biospecies. The biological species concept defines a species as members of populations that actually are potentially interbreed in nature, not according to similarity of appearance. Although appearance is helpful in identifying species, it does not define species. Organisms may appear to be alike and be different species. For example, western meadowlarks Sternella neglecta and eastern meadowlarks Sternella magna look almost identical to one another, yet do not interbreed with each other. Thus, they are separate species according to this definition. Organisms may look different and yet be the same species. 3. The ecological species concept defines a species as a set of organisms adapted to a particular ecological niche or lineage, or closely related set of lineages, which occupies an adapted zone minimally different from that of any other lineage in its range and which evolves separately from all lineages outside its range. 4. The phonetic species concept defines a species as a set of organisms that are sufficiently phonetically similar to one another. 5. The biological, ecological, and phonetic and several other species concepts are all closely related and are concerned to explain or describe much the same fact, that life seems to come in the form of distinct species. 6. Individuals mainly interbreed with other members of their own species because of isolating barriers that prevent interbreeding with other species. Isolating barriers can be prezygotic or postzygotic. 7. Geographic variation can be adaptive or neutral. The amount of genetic variation among geographic races of a species can be described quantitatively and is low in human beings relative to other species. 8. The theory of evolution justifies population thinking rather than typological thinking about interspecific variation. All individuals in a population are equally good members of a species, rather than some being better specimens than others. 9. Character displacement occurs when two species have partly overlapping geographic ranges and the two species differ more in symmetry than in allotropy. Character displacement may be caused by ecological competition. 10. The 10. The biological species concept explains the integrity of species by interbreeding, which produces gene flow, the ecological concept by selection. The two processes are usually correlated, but it is possible to test between them in special cases. Selection can be strong enough to overcome gene flow, and selection can maintain a species integrity in the absence of gene flow. 11. Taxonomic entities such as biological species may be real or nominal. According to the biological species concept, species can be real, but lower and higher taxonomic levels are nominal. According to the ecological species concept, all taxonomic levels can have a similar degree of realism. 12. Cladospecies, set of organisms between speciation events or between speciation event and extinction or a segment of a phylogenetic lineage between nodes. Upon speciation, the ancestral species is extinguished and two new species are named. 13. Evolutionary species is a unit of evolution or evolutionary group. A lineage and ancestral descendant sequence of populations evolving separately from others and with its own unitary evolutionary role and tendencies. Simpson. 14. Genetic species, group of organisms that may inherit characters from each other, common gene pool, reproductive community that forms a genetic unit. 15. Typological species, a group of organisms in which individuals are members of the species if they sufficiently conform to certain fixed properties. The clusters of variations or phenotypes within specimens, i.e., longer and shorter tails, would differentiate the species. This method was used as a classical method of determining species, such as with Linnaeus early in evolutionary theory. However, we now know that different phenotypes do not always constitute different species, e.g., a forming Drosophila born to a two-winged mother is not a different species. Species named in this manner are called morphospecies. 16. Morphospecies, species are the smallest groups that are consistently and persistently distinct and distinguishable by ordinary means. Contrary to the received view, this was never anything more than a diagnostic account of species. Morphospecies is a population or group of populations that differs morphologically from other populations. For example, we can distinguish between a chicken and a duck because they have different shape gills and a duck has webbed feet. Species have been defined in this way since well before the beginning of recorded history. This species concept was much criticized because more recent genetic data reveal that genetically distinct populations may look very similar and, contrarily, large morphological differences sometimes exist between very closely related populations. Nonetheless, most species known have been described solely from morphology. 16. Notospecies, species formed from the hybridization of two distinct parental species, often by polyploidy. 17. Phylospecies, the smallest unit appropriate for phylogenetic analysis, the smallest biological entities that are diagnosable and monophyletic, unit product of natural selection and descent. A geographically constrained group with one or more unique apomorphies, autapomorphies. 18. 19. Taxonomic species, specimens considered by a taxonomist to be members of a kind on the evidence or on the assumption they are as alike as their offspring or hereditary relatives within a few generations. Whatever a competent taxonomist chooses to call a species. 20. Ring species are species with a geographic distribution that forms a ring and overlaps at the ends. The many subspecies of Ensodina salamanders in California exhibit subtle morphological and genetic differences all along their range. They all interbreed with their immediate neighbors with one exception, where the extreme ends of the range overlap in southern California. E. cowrie and E. eschscholzii do not interbreed. So where do we mark the point of speciation? 21. Chronospecies are different stages in the same evolving lineage that existed at different points in time. Obviously, chronospecies present a problem for the biological species concept. For example, it is not really possible or very meaningful to figure out whether a trilobite living 300 million years ago would have interbred with its ancestor living 310 million years ago. 22. Phylogenetic evolutionary Darwinian species, a group of organisms that shares an ancestor, a lineage that maintains its integrity with respect to other lineages through both time and space. At some point in the progress of such a group, members may diverge from one another. When such a divergence becomes sufficiently clear, the two populations are regarded as separate species. 23. Microspecies, species that reproduce without meiosis or fertilization so that each generation is genetically identical to the previous generation. A2. Taxonomic terms. 1. Taxonomy is the science of naming and classification of organisms based on their genetic evolutionary relationships. Taxonomy runs very much a field of systematics in that it is based on phylogeny and comparisons between organisms and groups of organisms. It is the science of arranging living things into groups reflecting their relationships. E. Classification is the actual practice of placing animals into these groups. E. Nomenclature is the collection of rules and procedures of assigning names to the groups. 2. Higher taxa domain, kingdom, phyla, phylum, class, order, family, genera, genus, taxa, taxon, a taxon place. Taxa is a unit of classification of organisms. Higher taxonomic categories taxa group species together, one ideally according to what evolutionary relationships exist between species, just as human individuals group themselves into families, etc. Taxonomic categories, going from larger, most inclusive to smaller include 1. Domains. 111 phyla, phylum. B. Order. Build genera, genus. 1 kingdoms, 1 B class. B. Family, fifth species. 3. Artificial classification is based on one or a few easily observed characteristics, and is usually designed for a practical purpose with an emphasis on convenience and simplicity. The ancient Chinese system already mentioned is an artificial classification. Linnaeus included all worm-like organisms in a single group, the Burmese. This included a wide range of animals, from simple nematodes worms and earthworms to snakes. This was an artificial classification because it did not take account of important natural relationships, such as the fact that snakes have backbones and earthworms do not. Snakes have more in common with other vertebrates than with worms. An example of an artificial classification of fish could be to group them as freshwater fish, brackish water fish, and marine fish on the basis of their environment. This would be convenient for the purpose of investigating their mechanisms of osmoregulation. Similarly, all microscopic organisms are known as microorganisms, a convenient group for the purposes of study but not a natural group. 4. Natural classification tries to use natural relationship between organisms. It considers more evidence than artificial classifications, including internal as well as external features. It takes help from similarities of embryology, morphology, anatomy, physiology, biochemistry, cell structure, and behavior all relevant. It takes help from similarities of embryology, morphology, anatomy, physiology, biochemistry, cell structure, and behavior all relevant. Most classifications in use today natural and phylogenetic. 5. Phylogenies. A typical goal of systematic and paleontology is the construction of phylogonies. A phylogeny of a description of the genealogical circa, blood, i.e., evolutionary relationships between groups of organisms. A phylogeny thus can be a description of the macroevolutionary history of a species or more than one species. 6. A phylogenetic classification is one based on evolutionary relationship. In such a system, organisms belonging to the same groups are believed to have common ancestors. The phylogeny evolutionary history of a group can be shown by means of a family tree. 7. Phonetic classification. Another way to classify organisms wants to use a phonetic classification. This is an attempt to avoid the problem of establishing evolutionary relationships, which can be very difficult and very controversial, especially if there is little or no fossil evidence. The word phonetic comes from the Greek phenomenon, that which is seen. This classification is based solely on observable characteristics, phonetic similarity, and all characters used are considered of equal importance. All features of an organism can be considered, the more the better, and they do not necessarily have to. All features of an organism can be considered, the more the better, and they do not necessarily have to be of evolutionary significance. Masses of data are collected and the degrees of similarities between different organisms are calculated, usually by computer because the calculations are extremely complex. The use of computers in taxonomy is known as numerical taxonomy. Phonetic classifications often resemble phylogenetic classifications, but they are not constructed with this in view. 8. Cladogram. A cladogram is a graphical representation of a phylogeny. The cladograms come in a variety of types. Typical among all is an attempt to properly sort nodes, i.e., speciation events, such that descendant species are properly connected to their ancestral species, and species are grouped more closely to related species than they are to less well-related species. The is with the phylogeny it represents. The goal of a cladogram 1 is to properly represent correct evolutionary relationships. 9. Monophyletic taxon. Clade. The goal of systematics is to define monophyletic taxa, also known as clades. A monophyletic taxon is one that includes a common ancestor species as well as all descendant species. The ideal in systematics is for each taxon to be a monophyletic grouping, creating a classification that reflects the evolutionary history of organisms. Achieving this ideal is often easier said than done. 10. Polyphyletic taxa. A polyphyletic taxon represents a mistaken classification. Essentially, a polyphyletic taxon is one that contains at least two descendant species but not all ancestor species. Such things happen when a species once inadvertently included in a clade that it doesn't belong in. 11. Convergent evolution. Analogy. Polyphyletic taxa occur as a consequence of mistaken 10. Polyphyletic taxa. A polyphyletic taxon represents a mistaken classification. Essentially, a polyphyletic taxon is one that contains at least two descendant species but not all ancestor species. Such things happen when a species once inadvertently included in a clade that it doesn't belong in. 11. Convergent evolution. Analogy. Polyphyletic taxa occur as a consequence of mistaken analogies for homologies. Analogies are two structures that superficially resemble each other. 1. Which appear at the very least at first glance to be homologous but are not. Analogies result from convergent evolution. The two species do similar things in similar environments, so consequently evolve similar structures to perform these similar functions. The key difference between an analogy and a homology are twofold. The common ancestor between the two species will have lacked the common structure. The development of the structure will differ. More generally, homologies predict other homologies between two species, whereas analogies give rise to much less predictive power about the existence of additional homologies between two species. 12. Paraphyletic taxa. A paraphyletic taxon is also a mistake, but a legitimately made one, unlike a polyphyletic taxon. Like a polyphyletic taxon, a paraphyletic taxon does not represent a clade. However, paraphyletic taxa are not clades for a reason, and that reason has to do with evolutionary innovation. Typically, paraphyletic taxa are ones which include phenotypically similar descendant taxa, but exclude phenotypically dissimilar taxa. For example, reptiles form a paraphyletic taxon if mammals and birds are not included among the reptiles. Lizards are paraphyletic if you don't include snakes, which are derived from lizards. Similarly, the apes form a paraphyletic taxon if humans are not included among the apes. 13. Cladistics. Seed latistics is a technique by which organisms are assigned to different monophyletic. 12. Paraphyletic taxa. A paraphyletic taxon is also a mistake, but a legitimately made one, unlike a polyphyletic taxon. Like a polyphyletic taxon, a paraphyletic taxon does not represent a clade. However, paraphyletic taxa are not clades for a reason, and that reason has to do with evolutionary innovation. Typically, paraphyletic taxa are ones which include phenotypically similar descendant taxa, but exclude phenotypically dissimilar taxa. For example, reptiles form a paraphyletic taxon if mammals and birds are not included among the reptiles. Lizards are paraphyletic if you don't include snakes, which are derived from lizards. Similarly, the apes form a paraphyletic taxon if humans are not included among the apes. 13. Cladistics. Seed latistics is a technique by which organisms are assigned to different monophyletic taxa. Seed latistics works by identifying homologies and grouping together organisms such that within taxa individuals share more homologies than they do with individuals found in different taxa. Cladistics also rejects the inclusion of similarities that are the result of convergent evolution, i.e., analogies that are homologies but that are shared with other taxa. Note that this is not to say that it is necessarily easy to distinguish analogies from homologies. Cladistic techniques are not able to judge evolutionary divergence in terms of the time between nodes. Speciation events, time information instead is derived from the fossil record. Cladistic analysis has become synonymous with phylogenetic analysis. A clade clados, branch is an evolutionary branch. Cladistic analysis classifies organisms according to the order in time that branches arose along a dichotomous phylogenetic tree. Each branch point in a tree is defined by novel homologies unique to various species on that branch. Because it views the extent of divergence among organisms as uninformative in assessing evolutionary relationships, cladistic analysis considers only homologies in developing hypotheses about classification and phylogeny. 14. Shared derived characters. A cladis derives phylogonies from shared derived characters, aka synapomorphies, those homologies that are unique to individual taxa possessed by all members of a given clade. For example, in cladistics, the fact that bats have wings would not include bats in a taxa including birds on the basis of both having wings. Birds and bats both have wings as a consequence of convergent evolution, and a bat-bird taxon, were such a taxon to exist, would be a good example of a polyphyletic taxon since it would exclude the non-wing common ancestor to both the birds and the bats. In general, the existence of homologies predicts the existence of additional homologies, and cladistics uses these correlations to define taxa. 15. Shared primitive characters. Not all shared characters are shared derived. Characters for example, a cladist would not use the fact that both dogs and bears have hair as a means of classifying both dogs and bears as carnivores, since the ancestor of the ancestral carnivore, a mammal, also had hair. However, a cladist would use the fact that dogs and bears both have hair to include both among the mammals. Hair cannot be employed to distinguish mammals because hair is shared by all mammals, i.e., it is a shared primitive character when comparing among mammals, but a shared derived character when grouping mammals is distinct from other lineages. 16. Molecular systematics. In the past 2 decades, molecular systematics has come to dominate the study of evolutionary relations, which is not to say that molecular systematics has supplanted other approaches. It instead serves as an increasingly important tool. Molecular systematics seeks out homologies, like cladistics, though it is more difficult to distinguish analogies from homologies that ones, two nucleotides in a gene sequence could be identical because they have always been identical, i.e., since common ancestry, or due to divergence followed by mutation back to the same nucleotide. One advantage of this molecular tool of systematics is that it is objective and quantitative. A second advantage is that it can be used to assess relationships between groups of organisms that are so physiologically distant that they share very few morphological similarities. Third, molecular comparisons go right to the heart of evolutionary relationships. Molecular techniques include protein comparison. ddNA hybridization. D-restriction mapping and DNA sequencing. Biological NOMENCILATURE and theories of biological classification. Nomenclature. Botanical nomenclature is about naming plants. Bear in mind that plant names refer to abstract entities, a collection of all plants past, present, and future that belong to the same group. As you will recall, taxonomy is about grouping. Botanical nomenclature is about applying names to taxonomic groups. Scientific names of plants reflect the taxonomic group to which a plant belongs. One must first decide on the groups to be recognized. Only then does one start to be concerned about assigning an appropriate name to the plant. Scientific names are never misleading. No matter where you are, every plant has only one correct name. So long as its taxonomic treatment is not in dispute. This last one has a major reservation, but we can ignore it for now. The universality of scientific names means that even English-speaking people can find out what species grow in China or Saudi Arabia by reading a technical flora of these countries. Not only are the names the same, they are always written in the Latin alphabet, which is the same alphabet as these notes. Pronunciation. There is as little point about worrying over the correct pronunciation of scientific names as there is in worrying over which is the correct pronunciation of English words. Taxonomy refers to forming groups. Plants that belong to the same group have the same name. The taxonomic decisions concerning how a group is to be treated, what goes in the group, what rank it should be recognized as must be made before it can be assigned a name. It does. 4. Not matter how you decide what its affinities are unless, of course, you want others to support and use your treatment, but you must make these decisions before you can decide on an appropriate name for the group. So remember, taxonomy first. If people are going to communicate around the world, there needs to be an internationally accepted system of nomenclature. Creating such a system was not, and is not, an easy task. It was not until 1930 that agreement was reached on an international code had become standard around 1753. There were, however, many areas where there was widespread agreement in practice, with some of the practices dating back to before Linnaeus. For reasons that you will learn later, Linnaeus is taken as the starting point for botanical nomenclature. Let's consider for a moment some of the areas of agreement that existed before there was formal agreement on an international code of botanical nomenclature. The International Code of Botanical Nomenclature. Principles of botanical nomenclature. There are 6 principles that guide decisions concerning the code. 1. Uniqueness principle. Principle ID. The uniqueness principle states that there is only one correct name for a particular taxonomic group within a given taxonomic treatment. It is the central principle upon which all the remainder of the code is based. If people disagree on the taxonomic treatment, they will consider different names to be correct, but within any treatment, each taxonomic group has only one correct name. 2. Type principle. Principle 2. The type principle states the application of names of taxonomic groups is determined by means of nomenclatural types. For vascular plants such as grasses, a nomenclatural type is a herbarium specimen that has been deposited in herbarium. A nomenclatural type anchors the meaning of a name. If there is an argument as to what kind of plant the author of a name meant by a particular name, one examines the type specimen. No matter what taxonomic treatment is followed, the name must be used in a sense that includes its type specimen. If, as occasionally happens, the author of a new name provides a description that does not match the type specimen, it is the type specimen, not the description, that determines what kind of plant is called by the name in question. Adherence to the type principle did not become mandatory until 1958. Prior to that time, when taxonomists published a new name, they frequently simply listed several different specimens that exemplified what they meant by the name, without identifying any particular specimen as a top dog among the examples. All the designated specimens, including their duplicates, are referred to as syntypes, nomenclatural types of a single name, all of which were equally important. This became a problem if later taxonomists decided that there are 2 or more taxa among the specimens listed. When this happens, it became necessary to determine which of the specimens listed belongs with the original name. To prevent such situations arising, the rules for designating a type specimen were made more explicit. Since 1990, it has been necessary to identify the exact specimen that is to be the nomenclatural type of the taxon and the herbarium in which the specimen is located. Between 1958 and 1990, it was enough to specify who collected the specimen, where it was collected, the date on which it was collected, and the collection number it was given, if any. The problem was that, if the collector made several duplicate specimens, each of the duplicates is a syntype. In most instances, this is not a problem, but occasionally the supposed duplicates turn out to belong to different species. Requiring that an author state exactly which of the specimens is to be regarded as a nomenclatural type helps prevent even this kind of problem. If possible, the accession number of the type should be specified as well as the name of the herbarium in which it is located, but many older herbaria do not give their specimens accession numbers. There are several different kinds of type specimen, but the most important are holotypes, lectotypes, neotypes, and epitypes. The next most important are isotypes, syntypes, and paratypes. The first 4 kinds of type refer to specimens that are, unequivocally, the nomenclatural type of a name. A holotype is a specimen that has been designated the nomenclatural type of a name by the person creating the name. If the person who originally published a particular name did not designate a holotype, a later taxonomist may select a specimen to serve as the nomenclatural type. This specimen then becomes what is called the lectotype of the name. If the holotype or lectotype is destroyed or lost, a holotype is a specimen that has been designated the nomenclatural type of a name by the person creating the name. The first 4 kinds of type refer to specimens that are, unequivocally, the nomenclatural type of a name. A holotype is a specimen that has been designated the nomenclatural type of a name by the person creating the name. If the person who originally published a particular name did not designate a holotype, a later taxonomist may select a specimen to serve as the nomenclatural type. This specimen then becomes what is called the lectotype of the name. If the holotype or lectotype is destroyed or lost, a new type specimen can be selected. Such replacement types are called neotypes. An eptype 1 is a specimen selected to be the nomenclatural type of a name for which there is a holotype, lectotype, or neotype available. Why would it be necessary to select another specimen as a nomenclatural type? Sometimes the holotype, lectotype, or neotype simply does not show the features that are needed to determine, unequivocally, to which of 2 taxa it belongs. In such a case, it cannot be used to fix the meaning of a name. In such situations, another specimen can be selected as a anchoring specimen. It is this specimen that is the eptype. 3. Priority principle. Principle IIT. This principle states, in essence, that if a taxonomic group has been given 2 or more names, the correct name is the first name that meets the code standards for publication. Basically, this means that the priority of a name dates from the time that it was first published and made known to other botanists. Writing the name in a letter or email to a colleague does not count, nor do notes made on herbarium sheets. Taxonomic groups may end up with 2 or more names for several reasons. The most common reason is taxonomic disagreement, about which the code says nothing. Sometimes, the person publishing a later name is simply unaware that the group has already been named. In other cases, 2 or more names were given to different looking specimens of what was later treated as a single group. Whatever the reason, the priority principle states that only the first name validly and legitimately published for a particular taxonomic group is correct. In determining priority, the date that matters is the date on which the material was actually mailed to other institutions. This is not always the same as the year on the cover of a book or journal. 4. Retroactivity principle. Principle B. This principle states, the rules of nomenclature are retroactive unless expressly limited. The retroactivity principle means that anyone proposing a change in the code needs to consider the effect that the proposed change will have on names published in a wide range of literature and over a considerable period of time. This one's an intimidating requirement. The retroactivity principle means that anyone proposing a change in the code needs to consider the effect that the proposed change will have on names published in a wide range of literature and over a considerable period of time. This one's an intimidating requirement. It is why all proposed changes to the code undergo committee scrutiny before being voted on. If the committee has a problem with a proposed change, one of its members will get in touch with the person proposing the change. The committee member may point out unforeseen consequences of the proposed change. Alternatively, he or she may suggest examples that will make a stronger case for the change, or suggest modifications that will avoid some undesirable consequences. All proposals to change the code are published in Taxon, but they remain proposals until they are voted on at the next International Botanical Congress. 5. Principles 1 and B. The other 2 principles are straightforward. Principle 1 states that botanical nomenclature is independent of zoological and bacteriological nomenclature. If an organism is considered to be a plant, then it must be named in accordance with the botanical code. If it is considered a bacterium, it must be named according to the bacteriological code. Similar names for crop plants are given by ICNCP. Principle B states that scientific names are treated as if they were Latin, regardless of their derivation. Other key provisions of the code. 1. Any changes in the code should be designed to increase the stability of plant nomenclature. No one likes name changes, not even the taxonomists that propose them. 2. Every plant belongs to a species, every species to a genus, every genus to a family to an order, every order to a class to a division, also called a phylum nowadays, a concession to the greater number of zoologists in the world. This is the taxonomic hierarchy. Note that the code assumes the existence of species. It does not state what constitutes a species, let alone discuss whether species are real. The code also requires that plant diversity be summarized in a hierarchical structure. Again, it is not a question of whether such a structure really exists. The fact that the code assumes the existence of species in a hierarchical structure does not mean that the assumptions are correct, merely that, in naming plants and the zoological code is similar in this regard, one must act as if species are real in nature once hierarchical. Many people object to this, but no one has provided a persuasive argument for dropping the system. Publishing scientific names. Before a name, even a name that has a Latin form, can be accepted as a scientific name, it must satisfy several criteria. Some of these have to do with its form, others with how its existence and meaning are made known to others. Form. Principle B states that a scientific name must be treated as if it were Latin, but the Article 16 to 28 of the code also specify what form the name must take. I have summarized them in the table below. Rank ending examples. Division. Phylum Phytophytophyta, Magnoliophyta. Phenophyta, Liliophyta, class Ophyta, Magnoliophyta. Order Ales, Lilios, Magnolios family, Aciae, Pinaceae, Liliaceae. Y. Magnoliaceae, Tridiae, Pini, Liliae, Magnoliae. Genus Anand, Pinus, Lilium, Magnolia. Pinus flexilis, Lilium species depends Grandiflorum, Magnolia. Grandiflora variety depends Pinus flexilis var, Cumulus. Form depends. Family names must be formed by combining a generic name with a suffix, Aciae, but there are 8 exceptions to this rule. Each of the 8 exceptional names was almost universally used, and used in the same sense, throughout the world when the first edition of the code was prepared, and so, in accordance with the overriding goal of achieving nomenclatural stability, it was agreed that they would continue to be used. The 8 names are Brominiae, grass family, alternative Poaceae, palmae, palm family, alternatively Ericaceae, cruciferae, mustard family, alternatively Brassicaceae, Leguminosae, pea family, alternatively Fabaceae, guttiferae, St. John's wort family, alternatively Clusicaceae, umbelliferae, carrot family, alternatively Tilapiocaceae, labiatae, mint family, alternatively Lamiaceae, and compositae, daisy family, alternatively Asteraceae. The name of a species is always a binomial. Grandiflora is not the name of a species. It has to be combined with a generic name to form the name of a species, as in Magnolia grandiflora. The word grandiflora is what we call the specific epithet. It states which species of Magnolia is under discussion. Specific epithets are often adjectives that describe some attribute of the plant. It helps to learn a little Latin. Grandiflora means large flowered, but may refer to the habitat of a species, pregnances of fields, lacusters of lakes, saxicola, of rocky places, the place where the species occurs, chinensis, europaea, canadensis, or a person that is somehow connected to the species. The connection may be remote. Right, referring to a single male person named right, referring to a single female person named right. Right arm referring to 2 or more people, one of whom and possibly only 1 out of 100 was male, or right arm referring to 2 or more people with not even 1 male among them. The Romans were sexist. Technically speaking, subspecies is a higher rank than variety. A subspecies may include several varieties. In practice, most taxonomists nowadays use one rank or the other, but not both. Europeans tend to use subspecies and expect subspecies to occupy somewhat different areas, whereas Americans. Technically speaking, subspecies is a higher rank than variety. A subspecies may include several varieties. In practice, most taxonomists nowadays use one rank or the other, but not both. Europeans tend to use subspecies and expect subspecies to occupy somewhat different areas, whereas Americans use variety to denote plants that are different from the plants first put in the species. In practice, the 2 ranks are used almost interchangeably. Writing scientific names. In North America, it is customary to write names at the rank of genus and below in italics or some other font that sets them apart from the rest of the text. The most recent edition of the code recommends that all scientific names, no matter what their rank, be in a different font from the rest of the text. Either practice makes it easy to scan for taxonomic information. The names of all ranks from subgenus up must be capitalized. In most instances, the specific epithet and epithets for lower rankings must not be capitalized. There are some exceptions to this rule, cases where it is permissible, but not required, to capitalize a specific or real epithet, but you need to be careful. Personally, I recommend always using lowercase for epithets, names distinguishing species in lower ranks. That way one is never wrong. Authorities. You will notice that scientific names are often followed by word or a capital letter and a period, or one or more unintelligible to the uninitiated sets of letters. To join the initiated, read on. The letters and or words that follow a scientific name, sometimes they may be with a name more on that later, or a shorthand reference to the name of the person that first gave the name to the entity involved in some instances, to the persons who first treated it at the rank being used. This is probably easier to understand through some examples. Consider Oryzopsis exigua thurber. Note that only the first 2 words are italicized. This means you are looking at the name of a species. Thurber is the last name of the person who first gave a name to this species, and the name he gave to it is the one shown. Consider Oryzopsis aspergillomycetes. Again, you are looking at the name of a species in the genus Oryzopsis. This species was first named by a fellow whose name is abbreviated to Mycetes. The period tells you that his name has been abbreviated. His full name was Michaud. To whom do you think L refers to in Triticum estivum L? Dicanthelium lanugginosum Elliott Gould. The name is Dicanthelium lanugginosum. As you immediately recognize, because the name is a binomial, the entity being named is being treated as a species. The first person to give a name to this species was a chap whose last name was Elliott, but he named it Panicum lanugginosum. An inner circle of initiates could tell you that Elliott refers to Walter Elliott, who lived from 1803 to 1887. In eastern North America, there is a book called Authors of Plant Names that provides such insight. Gould stands for Frank W. Gould came along later and decided that, although Elliott was right in describing the species, he should have put it in a different genus, the genus Dicanthelium. Elliott's name is in parentheses to show that he was the first person to say, aha, these plants are different. Gould's name is outside the parentheses because he said, yes, Elliott was right, these plants are different, but they should be included in the genus Dicanthelium, not Panicum. Consider Disticus spicata L. Green. Linnaeus L stands for Linnaeus first described the entity, but it's Uniolus spicata, not Disticus spicata. Green was the first person to say no, these plants should be in Disticus and then published a combination, Disticus spicata. Linnaeus gets credit for being the first person to describe the entity, Green for being the person to give it the name shown. Most journals, and consequently many professors, ask that you cite the authorities for a name when it is first used. It is a rather meaningless exercise. It is meant to say, I am using this name in the sense that it was used by Green in the last example, but really you are probably using it in the sense that it is used in some flora based on what your boss told you. The 1999 Congress encouraged editors to be more rational about when it was useful to cite authorities and when not, but I suspect that most journals will continue to require them for some time to come. Proposing a new name or new combination. If you have to publish a new name or combination, the code requires that you follow certain rules, which it calls articles. The key requirements are that equals the new name or combination be published in a normal botanical outlet, not the Herald Journal or Statesman, copies of which are sent to at least 2 botanical institutions. Equals if the name is for a new taxon, the distinguishing characteristics of the taxon, and preferably a full description, must be given in Latin and a holotype specified. Equals if the name is simply a new combination, perhaps reflecting the transfer of a species from one genus to another or its demotion to a subspecies, there must be a clear and complete reference to the place where the original name was first published. Structural biochemical and molecular systematics. Systematics. Systematics 1 is the study of relationships between organisms. It includes all relationships between organisms in an attempt to identify patterns in their similarities. Systematics includes those areas of biology concerned primarily with similarities and differences between organisms. It includes disciplines such as evolution, phylogeny, comparative anatomy, comparative physiology, comparative biochemistry, comparative embryology, comparative ecology, paleontology, taxonomy, and classification. The object of systematics is the uncovering of genetic relationships that will shed light on the evolution, or phylogeny, of the organisms being compared. Classification. There are between 1.4 by 10 degrees, known as the 1992, and 100 by 10 degrees maximum estimate species alive on the planet today. With this many organisms, we must have a phyling, or classification, system so we can keep track of the species, their relationships, and what we know about them. In biology, we use a classification system based on genetic relationships. The system reflects the evolution of the organisms classified. For example, consider cats, tigers, humans, flies, oaks, and ammonia bacteria. We would use systematic biology, including anatomy, behavior, physiology, and biochemistry to group these organisms according to their degree of kinship. We use physical similarity as our index to kinship and assume that similar organisms are more close related than dissimilar ones. We can construct a simple phylogeny, or phylogenetic tree, showing the relationships between these 6 organisms using our current knowledge of their appearance in cell biology. The resulting diagram is a cladogram in which the vertical axis is time and the branching points show the time of the most recent common ancestor between 2 groups. We will build the cladogram in class beginning with cats and tigers because they seem to be the most closely related of any pair of organisms in the collection. We then arrange other organisms on the cladogram according to how closely related to cats they seem to be. The classification system proposed in 1758 by Carl Linnaeus reflects these evolutionary relationships and is a system used by all biologists today. In its original form, it included 7 categories, ranks, but we have added many more. Each of the world's known organisms is positioned in a hierarchy of categories with other organisms to which it is related. Thus, those with very recent common ancestors and little time to diverge are grouped closely together, and those with more remote ancestors are placed in different groups. The most closely related are placed in the same species. The species is the most restrictive and least inclusive category. Membership in the same species means the organisms share a very recent common ancestry and have had very little time to diverge, hence they are very much alike. All humans belong to the same species, Homo sapiens, and while we are not identical, we are very much alike. At the other end of the hierarchy is the kingdom. This category is the most inclusive and least restrictive. To belong to the same kingdom as another organism does not require recent common ancestry, genetic commonality, or very much similarity. Humans and jellyfish belong to the same kingdom, Animalia. For example, a Linnean classification of a human would be as follows. A category is a level in the following hierarchy. Kingdom in order of categories. A taxa plural. Taxa is a unit in any category. Reptilia, Homo, and Annelida are taxa. Linnaean classification of humans. Superkingdom, Eukaryota plus plantae, fungi. Protoctista kingdom, Animalia plus Cnidaria, Annelida. Mollusca, Arthropoda, etc. Phylum, Chordata plus Cephalochordata. Urochordata subphylum, Vertebrata plus Osteopods, abs. Reptilia, etc. Class, Mammalia plus Carnivora, Rodentia. Parasitactyla, etc. Order, Primata plus Hominidae, Homidae, etc. Family, Hominidae plus Homo. Australopithecus, Zygoanthropus, etc. Genus, Homo plus H. habilis, H. erectus, etc. Species, Homo sapiens. Of these categories, only the species is real and exists in nature. The others are artificial constructs we make for our own convenience and are not unambiguously identifiable in nature. A species, at least in sexually reproducing organisms, is defined as being a group of potentially interbreeding individuals that cannot interbreed with individuals outside the group. Nomenclature. Every species, when it is first described by a taxonomist, is assigned a binomial name. This name consists of the generic name, which is always capitalized, and the specific epithet, which is not. Thus our species is Homo sapiens. When written, the scientific name is placed in a different type font, usually italics, than the rest of the text that accompanies it. The scientific name is unique and no other organism in the kingdom can have one it. Phylogeny. One concern in this series of lectures is with phylogeny, how it is reconstructed, and how our knowledge of it is incorporated into our classification of organisms. Our understanding of phylogeny is based in part on fossils. A fossil is any preserved remnant of an organism that lived in the past. Most organisms die without leaving a trace, but occasionally, rarely, some aspect of the organism may be preserved for great lengths of time, sometimes millions of years. Fossils may be actual hard parts of former organisms. They may be casts of organisms or their parts. They may be preserved artifacts made by the organism such as burrows or footprints or fecal pellets. Many fossils are not the actual parts of the original organism but replacements that have been mineralized. The fossil record is very incomplete for a number of reasons. The discovery of a fossil by a paleontologist is a culmination of a series of improbable events. First, very few organisms die under circumstances that make fossilization likely. Relatively few individuals become fossils to start with. Second, the fossils must survive millions of years of geologic events such as erosion, volcanism, pressure, faulting, orogenese, mountain building episodes, earthquakes, etc. and remain intact. Thirdly, the fossil must occur or be moved by some force to a location or near the ground surface where it has a chance of being found. Finally, the fossil must be noticed by someone who recognizes it as being worthy of attention. All of these steps are improbable. Dating. For fossils to be useful in constructing phylogenies, we must have some idea of their age. We use relative and absolute dating. Relative dating. Most fossils are formed in sediments laid down in water. These sediments become sedimentary rock, which then may be lifted above the water to become dry land during mountain building episodes. The sediments and the fossils are laid down in chronological order, with the oldest in the lower layers of strata and the youngest in the upper strata. Simply knowing the level of the stratum of a fossil gives information about its age relative to other fossils in associated strata. We also have methods of absolute dating whereby we can put actual ages, in years, on fossils. Most absolute dating methods rely on known rates of radioactive decay whereby a radioactive isotope decays to a stable isotope. Carbon-14, for example, has a radioactive decay rate such that 1T takes 5,600 years for half of it to decay to nitrogen. 1C and C are taken up by living organisms in the ratio present in the environment. Let this initial ratio be x. Once the organism dies, the asterisk C begins to decay at its known rate. In 5,600 years, the ratio will be x2. In 11,200 years, it will be x4, and so on. By measuring the amount of 1C remaining in carboniferous fossils and comparing the ratio with that originally present. Because of its relatively short half-life, C is useful only for times less than about 50,000 years. After that time, there is not enough C left to measure accurately. Carbon-14 consequently is very useful in studies of recent human prehistory. Other isotopes, such as potassium-argon, are used for longer time ranges. Potassium-40 has a half-life of 1.3 billion years. Macroevolution is evolution above the species level. It is the evolution of higher taxonomic categories such as genera, families, orders, classes, and phyla. Does it occur by the same mechanisms as microevolution but over larger periods of time? Evidence for phylogeny. For the first 200 years of the study of biology, physical anatomical similarity was the basis for almost construction of phylogenies and classifications. Early biologists relied on anatomic similarities, fossil similarities, and embryological similarities, all of which are anatomic, to determine which organisms were closely and which distantly related. Our early understanding of evolution was based on anatomy. Recently, however, new and powerful sources of evidence have been provided by molecular biology and biochemistry. We can now look at amino acid sequences of proteins or at the genes encoding those proteins for similarities or differences. This is almost like having the answers at the back of the book. We have struggled mightily to discover the true genetic relationships between organisms using anatomy, and now we can verify our ideas with an independent and very powerful new set of data. Neither method is infallible, and we should continue to use both and check the results of each against the other. The new molecular techniques are based on the assumption that closely related organisms should have proteins with very similar amino acid sequences, or, even better, genes with the same or similar base sequences. This follows the same reasoning developed earlier. The more time that has elapsed since the last common ancestor, the more time there has been for differences to develop. Protein sequences. As you know, the amino acid sequence of a polypeptide is determined genetically in that it once encoded in the nucleotide sequence of the gene. Genes undergo point mutations regularly, and if the mutations have no discernible effect on the protein, will persist in the genome indefinitely. Thus, nucleotide sequences would be expected to change as time passes and point mutations accumulate. Comparison of the primary structures of the same protein from different organisms can tell us how similar the genes for that protein are in the two organisms. From that we can deduce how much change has taken place since the two organisms shared their last common ancestor. Cytochrome scent, for example, is a polypeptide of 10 inches of amino acids present in all anaerobic organisms in association with an electron carrier in the electron transport chain on the mitochondrial cristae. Its amino acid sequence has been determined for a wide variety of representative organisms. If you were asked to use anatomic characteristics to arrange a rhesus monkey, tuna fish, rattlesnake, chimpanzee, and dog according to how closely related to humans each is, you would probably produce the following: chimp, monkey, dog, snake, fish, and this is exactly the arrangement biologists have agreed upon for centuries. The following data are from amino acid sequencing studies of cytochrome circa. Humans and chimpanzees have identical cytochrome scent molecules. All 104 amino acids are the same. Humans and rhesus monkey have a difference of 1 amino acid out of 104. Humans and dogs have 13 different amino acids. Humans and snake have 20 different amino acids. Humans and tuna fish have 30 different amino acids. A phylogeny made using cytochrome C is identical to the traditional one using anatomy. DNA hybridization. It is now possible to compare the genes of two organisms directly to see how similar their nucleotide sequences are. One technique for doing this is DNA hybridization. In this method we look, not at the gene product protein, but at the gene itself. DNA from two organisms of interest is removed and heated to cause the two strands to separate into complementary strands. The single-stranded DNA from the two species is mixed and allowed to cool so that double-stranded DNA will reform. The new double strands are hybrids. That is, they are composed of one strand from each species. How tightly the two strands bond to each other is a measure of how similar their nucleotide sequences are. If they are perfect complements, with no different nucleotides, they will bond perfectly at all base pairs for the entire length. If they differ, they will not find partners for all their bases. The more different they are, the poorer the match and the looser the bonding. The hybrid molecules can be heated to cause them to separate again. The amount of heat needed to separate them is proportional to the tightness of the bonding between them. If the nucleotide sequences are identical, then more heat will be required. The more different they are, the less the heat that will be needed. If we construct our phylogeny correctly, using only homology and no analogy, the resulting taxa are monophyletic, meaning they have one common ancestor. If we slip up and include some analogy, as in our tuna example, the resulting group is polyphyletic, since there are many unrelated ancestors. Tuna, sharks, ichthyosaurs, and porpoises do not have a single common ancestor. A group is polyphyletic if its members are derived from two or more ancestors that are not common to all. Sometimes we create polyphyletic groups knowing we are probably doing so, but we lack enough information to make monophyletic groups. The Protoctista, for example, is recognized as a polyphyletic group of many very different organisms that have little in common other than their unicellularity. As we learn more about these organisms, we gradually remove them from the Protoctista and place them in monophyletic groups. Green algae, brown algae, red algae, diatoms, flagellates, ciliates, amoebas, others. Numerical taxonomy. In 1963, Sokol and Sneath, Sokol, R.R. and Sneath, PhD 1963 Principles of Numerical Taxonomy, San Francisco introduced the new concept of numerical taxonomy. This new approach to taxonomy employs computers and statistical methods for establishing the degree of similarity among taxa. They laid down certain postulates which in their own words are as follows. 1. The greater content of information in the taxa of a classification and the more characters on which it is based, the better a classification will be. 2. A priori every character is of equal weight in creating natural taxa. 3. Overall similarity between any two entities is a function of individual similarities in each of the many characters in which they are compared. 4. Distinct taxa can be recognized because correlations of character differ in the groups of organisms under study. 5. Phylogenetic inferences can be made from the taxonomic structures of a group and from character correlations given certain assumptions about evolutionary pathways and mechanisms. 6. Taxonomy as viewed in practice is an empirical science. 7. Classifications are based on phonetic similarity. In numerical taxonomy, it is the computer and not the author who fixes the weightage for the characters. Therefore, the subjective element has yielded place to the objective, and this is the greatest merit of numerical taxonomy. On par with its merit of objectivity, there's also the other merit of being able to handle hundreds of unique characters of extremely diverse kinds taking the help of a computer. From the foregoing account, it follows that a numerical taxonomist has to proceed through the following steps for the realization of his goals. 1. Choice of units for study. Whether it is an individual or strain or species that is to be the subject of study is first determined. The smallest units figuring within a program of study are called operational taxonomic units (OTU). 2. Character selection. Each OTU is carefully studied for determining its unit characters, a unit character being one or two or more states, not lending itself for further subdivision. For a fairly satisfactory analysis, at least 50 to 100 such unit characters should be determined. 2. Character selection. Each OTU is carefully studied for determining its unit characters, a unit character being one or two or more states, not lending itself for further subdivision. For a fairly satisfactory analysis, at least 50 to 100 such unit characters should be determined. Once the unit characters are fixed, they are coded. In a two-state coding, the characters are divided into plus and whereas 1 and O. The positive characters are recorded as plus, whereas 1 and negative characters is worth 0.1 in case the organ possessing a given character is missing in an organism. The character must be scored NC, which means no comparison. The multi-state characters may be either quantitative or qualitative. Quantitative characters can be expressed by a single numerical value corresponding to the range of variation. Qualitative multi-state characters cannot be arranged in some order, and no reliable sequence can be established. 1. Choice of units for study. Whether it is an individual or strain or species that is to be the subject of study is first determined. The smallest units figuring within a program of study are called operational taxonomic units (OTU). 2. Character selection. Each OTU is carefully studied for determining its unit characters, a unit character being one or two or more states, not lending itself for further subdivision. For a fairly satisfactory analysis, at least 50 to 100 such unit characters should be determined. Once the unit characters are fixed, they are coded. In a two-state coding, the characters are divided into plus and whereas 1 and O. The positive characters are recorded as plus, whereas 1 and negative characters is worth 0.1 in case the organ possessing a given character is missing in an organism. The character must be scored NC, which means no comparison. The multi-state characters may be either quantitative or qualitative. Quantitative characters can be expressed by a single numerical value corresponding to the range of variation. Qualitative multi-state characters cannot be arranged in some order, and no reliable sequence can be established. In such cases, qualitative characters are conveniently converted into some new characters. The information secured is used for preparing ETXN table, T standing for the number of OTU and N for the number of unit characters. This table gives us a data matrix. The table below is a data matrix for 4 OTS and 3 unit characters. Characters taxa OOTU. A, B, C, D. 1 plus plus NC 2 plus 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 3 plus plus plus. 3. Measurement of resemblance. From the data so collected, a comparison of the OTU is made and the degree of similarity is established. If the similarity is complete, it is then expressed as S 100% similar, and if there is no resemblance whatever, the similarity is 0, 0. From this data, a similarity table, tabulating the S coefficients, 1 for each OTU is then constructed, which enable us to bring together all those OTU whose members have the highest mutual similarity. These constitute clusters or phenons. 4. Cluster analysis. The next logical step is an analysis of the clusters. We may fix arbitrarily certain levels of similarity as appropriate to certain ranks and on the basis of S coefficients running through the members of the cluster. Each members may be assigned a rank appropriate to it in terms fixed earlier. By arranging the various OTU into the respective clusters based on their appropriate ranking, we may draw different phenon lines, each indicating the degree of closeness between the OTU under study. 5. Discrimination. Once this study is completed, the entire data matrix will show us which characters are more constant and which therefore should be employed for Kenan diagnosis. Classification of bacteria. The main aim of bacterial taxonomy is to set up a classification and ordering of bacteria into groups based on common properties. This classification can then be used to identify individual bacterial species or strains, identification, and name of nomenclature. Classification is also used to study relationships of different bacterial groups based on shared phenotypic properties and probably a common evolutionary history. A wide range of morphological, biochemical, and molecular characters are measured, and the information derived from this is used to create a hierarchical classification system into which individual bacteria can be placed. The most important rank in the taxonomic hierarchy is called the species, which from the time of Linnaeus 1707-1778 have been named in a similar way. Related species are grouped into a genus place. Genera, which is usually based around some distinct phenotypic character, and the generic name is used as part of a binomial species name. So, for example, Escherichia coli is in the genus Escherichia named after Escherick, who first isolated the bacteria in 1885. The specific epithet, coli, is in this case an indication as to where the organism dot was isolated from, i.e. the colon. However, note that the binomial name is a label, not a description. It does not have to describe the species in any way. Note too that the genus name has a capital initial letter, but the specific epithet, which cannot be used alone, does not, and that the whole binomial name is Latinized and therefore should be italicized or underlined. Normally, once the name of the organism has been written in full, the genus name can be reduced to a letter or letters as long as there can be no confusion with other genera. Thus, Escherichia coli will subsequently be written as E. coli. The higher taxonomic categories into which the organism can be ranked are sequentially, family, order, class, division, and kingdom, though the final three are not particularly useful in bacteriology. Similarly, species may be divided into subspecies, and a culture of a particular organism is usually referred to as a strain. Strain is also used to describe a subgroup of a species that shares some features such as one type of antigen or mutation. There are also informal names, not italicized, which are sometimes useful for describing groups of organisms such as the green sulfur bacteria or the spirochetes. The main impetus for bacterial classification has been the need to be able to identify bacterial disease-causing organisms, and therefore these microbes are particularly well classified. However, it should be remembered that many other species have not been well characterized and many have not been isolated or described at all. The authority on bacterial classification is Bertie's Manual of Systematic Bacteriology, which contains as nearest possible complete listings of all prokaryotic species and their distinguishing characteristics. Characters used in bacterial identification. A list of the types of characters used to distinguish between bacteria is given below. This is not an exhaustive list, and equally not all characters are used in either the classification or identification of every bacterial species. In many cases of well-known microorganisms, a small set of tests may be all that is required to identify a microbe positively, especially if the site from which it has been isolated and the disease it caused are known. Akama-shaped, gram-negative rod isolated from a patient excreting copious amounts of straw colored liquid stools may be enough to identify it putatively as Vibrio cholera, the causative agent for cholera. However, it may be necessary to do a wide range of tests to determine the name of an isolated if it is an undescribed microbe. A. Colony morphology. The shape, texture, and color of bacterial colonies can be distinctive. For example, Staphylococcus aureus is so named as colonies are of a yellow color. Aureus comes from the Latin for golden. B. Cell shape, structures, and reaction to stains. Whether bacteria are gram-positive or gram-negative is one of the most important clinical diagnostic tests. Gram staining and microscopic examination of bacteria deformation as to the shape, size, and arrangement of the bacterial cells, and in some cases this is enough to identify certain genera such as streptococci, which are gram-positive cocci that grow in chains. Stains may also be used to show other morphological features, which may use for classification, such as the presence of equals force malachite green stain. Institute for Advanced Studies, Job 11. Equals unusual cell walls, Ziehl-Neelsen acid fast equals flagella, flagellar stain. Stain equals metachromatic granules, Albert equals capsules, Indian ink stain, Laybourne stain. Equals intracellular lipids, Sudan black. Hydrogenobacteria aquifex, pyrophyllus, rhomodales, Rhodobacterium icelandicum, Dermatoga maritima. Chioroflexus aurantiacus. Green non-sulfur Dermomicrobium roseum. Dermomicrobium thermophilus, Deinococcus radiodurans. Cyanobacteria senecococcus CIF 6301 Neisseria gonorrhea J. Rhodocyclus gelatinosus. Pseudomonas aeruginosa J. Escherichia col. Chromatium venosum rickettsia type. Rhodospirillum rubrum. Deinococcus group. Rhodobacter capsulius. Purple bacteria Agrobacterium tumefaciens and relatives Wallenella genes. Mixococcus xanthus Vitello vibrio stoipi. Desulfuromonas acetoxidans. Chionidia sitaki. Planktomyces E. Planktomyces stallidi. Trichomema pallidum. Spirochetes Spirocheta halofita. Serpula hydrodysenterii. Leptoma linei green sulfur chlorobium limicola. Cytophagus flexibacter flexilis. Cyprospira grandis bacteroides fragilis. Cytophaga iotica. Mycobacterium tuberculosis. Propionibacterium acnes. Micrococcus luteus. Streptomyces lididins J. Gram-positive lactobacillus minutus L. Bacteria megasphaera alzini. Heliobacterium florum Clostridium pastreum J. Clostridium aminovalerium Clostridium dermosicarolyticum J. _. Mycoplasma pneumoniae. Mycoplasma capriculum_ill. Acaleplasma labiali. Clostridium ramosum. Bacillus subtilis. Lactobacillus lactis. Lactobacillus casei. Leuconostoc mesendroids. Figure 2. A phylogenetic tree of bacteria derived from 165 ribosomal RNA sequences. Reproduced from Madigan et al. Raw Biology of Microorganisms, 7997, with permission from Prentice Hall, Inc., Upper Saddle River, NJ. 12 Institute for Advanced Studies, Jodhpur. C. Growth characteristics, the temperature, pH and O, requirements of isolate are useful tests for the identification of bacteria. D. Biochemical tests. There is a wide range of tests available that measure various aspects of bacterial metabolism. Equals what carbon and nitrogen sources the bacteria can use, equals the end products of their metabolic processes, such as acetylcholine, tested from the Biot Proscator test. Equals what enzymes the bacteria produce, such as decarboxylases, proteases and DNases, equals the presence of other molecules such as toxins, long-chain fatty equals acids or antibiotics. E. Immunological tests. Antibodies to cellular components such as the oside chains of lipopolysaccharide, LPS, or capsules are frequently used to distinguish between strains of one species. F. DNA tests. Comparisons of DNA content and sequence between strains are the most definitive methods for separating organisms into different groups and for measuring the evolutionary relatedness between them. Phylogenetic analysis. Eubacteria. Traditionally, DNA was analyzed by measuring the GC content or the amount of DNA homology between strains. More recently, the comparison of the actual DNA sequences of conserved cellular molecules such as 16S ribosomal RNA or proteins such as cytochrome C, ATPase and elongation factor has been used. This information has been analyzed by a technique called cladistics to trace through phylogenetic lineages that have not been obscured by patterns of convergent evolution. Current classification of bacteria. On the basis of 16S RNA sequences, it is now clear that there are at least 3 domains of organisms, the 2 prokaryotic groups. Archaebacteria and Eubacteria and Eukarya pig. 1. Within the Eubacteria there are at least 11 groups, referred to sometimes as phyla, which can be represented on a phylogenetic tree. Figure 2. What can be seen as a wide diversity within the bacterial world and that many of the bacteria that may be familiar to us such as E. coli, Bacillus species and Neisseria gonorrhoeae are located within just a few of these phyla. It should be noted that the number of phyla will increase as more bacteria from diverse habitats are sequenced. ARCHAEBACTERIA Eukarya. Green. Animals non-sulfur slime. Bacteria Entomobae modes gram. Methanosarcina methanobacterium. Methonococcus. Fungi. Halophils plants ciliates. Purple. Bacteria Dermoproteus dermococcus cellular cyanobacteria pyridium trichomonas flavobacteria. Dermovidales. Microsporidia. Diplomonids. Figure 1. An overview of the phyogeny of the living world, showing the 3 major domains of organisms. Institute for Advanced Studies, Jodhpur 13. Tissues, organs, and organ systems. Cells are the basic building blocks of multicellular animals. When cells with the same characteristics or specializations are grouped together, they form a tissue. There are 4 basic types of tissues: epithelial, connective, muscle, and nervous, but there are variations on each basic type. An organ is usually made up of several different tissue types. Figure 1. 1. 4 types of tissue. Tissue type and function. An organ is composed of tissues. Epithelial tissue. Lining, transport, secretion, and stomach absorption. Connective tissue support, strength, and elasticity. Muscle tissue movement. Nervous tissue information. Synthesis. Communication, and control. Within an organ, tissues are organized in specific ways. Epithelial tissues cover the body and line organs. Epithelial tissues are sheets of densely packed, tightly connected cells that cover inner and outer body surfaces. They form the skin and line hollow organs of the body, such as the gut. Figure 2. Some epithelial cells have secretory functions. Examples of the groups of epithelial cells that secrete hormones, milk, mucus, digestive enzymes, or sweat. Other epithelial cells have cilia to help substances move over surfaces or through tubes. Since epithelial cells create boundaries between the inside and the outside of the body and between body compartments, they frequently have protective as well as absorptive and transport functions. Epithelial cells can also form receptors that provide information to the nervous system. Smell and taste receptors, for example, are epithelial cells that detect specific chemicals. Epithelial tissues have distinct inner and outer surfaces. The outer surface faces the air, as in the case of the skin and lungs, or a fluid-filled organ cavity, such as the lumen of the gut. These outer surfaces are the apical ends of the epithelial cells, which may have cilia or may be highly folded to increase their surface area. The inner surfaces of an epithelium are the basal ends of the epithelial cells, which rest on an extracellular matrix called the basal lamina. The skin and the lining of the gut are examples of epithelial tissues that receive much wear and tear. Accordingly, cells in these tissues have a high rate of cell division to replace cells that die and are shed. Dandruff consists of discarded skin cells. Connective tissues support and reinforce other tissues. In contrast to densely packed epithelial tissues, connective tissues consist of dispersed populations of cells embedded in an extracellular matrix that they secrete. The composition and properties of the matrix differ among types of connective tissues. An important component of the extracellular matrix secreted by connective tissue cells is protein fibers. The dominant protein in the extracellular matrix is collagen. See figure 4.26. Collagen is, in fact, the most abundant protein in the human body, representing 25% of total body protein. Collagen fibers are strong. They give the connective tissue of skin, tendons, and ligaments resistance to stretch. Similarly, collagen fibers provide a net-like framework for organs, giving them shape and structural strength. Connective tissue that fills spaces between organs has a low density of collagen fibers. Another type of protein fiber in the extracellular matrix of connective tissues is the stretchable protein elastin. It can be stretched to several times its resting length and then recoil. Fibers composed of elastin are most abundant in tissues that are regularly stretched, such as the walls of the lungs and the large arteries. Gradual loss of elastin fibers with age causes gradual loss of resiliency of the skin. Two epithelial tissue. A single layer of cuboidal cells forms a simple epithelium lining the collecting ducts of a human kidney. B multiple layers of squamous cells form a stratified epithelium of cuboidal cells. In simple epithelium. 14 Institute for Advanced Studies, Jodhpur. Cartilage and bone are connective tissues that provide rigid structural support. In cartilage, a network of collagen fibers are once embedded in a flexible matrix consisting of a protein carbohydrate complex. Cartilage, which lines the joints of vertebrates, is resistant to compressive forces. Since it is flexible, it provides structural support for flexible structures such as external ears and noses. The extracellular matrix in bone also contains many collagen fibers, but it is hardened by the deposition of the mineral calcium phosphate. Adipose tissue is a form of loose connective tissue that includes adipose cells, which form and store droplets of lipids. Adipose tissue, or fat, is a major source of stored energy. It also serves to cushion organs, and layers of adipose tissue under the skin can provide a barrier to heat loss. Blood 1 SA connective tissue consisting of cells dispersed in an extensive extracellular matrix, the blood plasma. The blood plasma is much more liquid than the extracellular matrices of the other connective tissues, but it too contains an abundance of proteins. Muscle tissues contract. Muscle tissues consist of elongated cells that can contract and cause movement. Muscle tissues are the most abundant tissues in the body, and when animals are active, they use most of the energy produced in the body. Nervous tissues process information. There are 2 basic cell types in nervous tissues, neurons and glial cells. Neurons, which are extremely diverse in size and form, communicate via electrochemical signals. These nerve impulses can be conducted via long extensions of the neurons to other parts of the body, where they are communicated to other neurons, muscle cells, or secretory cells. Neurons are involved in controlling the activities of most organ systems to achieve homeostasis. Glial cells do not generate or conduct electrochemical signals, but they provide a variety of supporting functions for neurons. There are more glial cells than neurons in our nervous systems. Organs consist of multiple tissues. A discrete structure that carries out a specific function in the body is called an organ. Examples are the stomach, the heart, the liver, and the kidney. Most organs include all 4 tissue types. The wall of the stomach is a good example. The inner surface of the stomach that contacts food one's line with a sheet of epithelial cells. Some of the epithelial cells secrete mucus, enzymes, or stomach acid. Beneath the epithelial lining is connective tissue. Within this connective tissue are nerves, glands, clusters of secretory epithelial cells, and blood vessels. Concentric layers of smooth muscle tissue enable the stomach to contract to mix food with digestive juices. A network of neurons between the muscle layers controls these movements and also influences the secretions of the stomach. Surrounding the stomach is a sheath of connective tissue. An individual organ is usually part of an organ system, a group of organs that function together. The stomach is part of the digestive system, which also includes the food tube, esophagus, the small and large intestines, the pancreas, which secretes digestive enzymes, and the liver, which secretes bile. The major organ systems of mammals are outlined in Table 1. One of the major organ systems of mammals System tissues and organs functions. Nervous system, brain, spinal cord, sensory. Organs, peripheral nerves, endocrine system, glands, pituitary, thyroid, parathyroid, pineal, adrenal. Testes, ovaries, pancreas. Skeletal muscle, smooth muscle. Cardiac muscle. Muscle system. Skeletal system, bones, reproductive system. Vagina, mammary glands. Male, testes, sperm ducts. Accessory glands, penis, digestive system, mouth, esophagus, stomach, intestines, liver, pancreas. Rectum, anus. Respiratory system, airways, lungs, diaphragm, circulatory system, heart and blood vessels. Lymphatic system, lymph and lymph vessels, lymph nodes, spleen. Immune system, many types of white blood cells, skin system, skin, sweat glands, hair. Excretory system, kidneys, bladder, ureter, urethra, female, ovaries, oviducts, uterus. Receives, integrates, stores information and controls muscles. And glands, a system of glands releases chemical messages, hormones that control and regulate other tissues and organs. Produces forces and motion. Provides structural support for the body. Produces sex cells and hormones necessary to procreate and nurture offspring. Acquires and digests food, absorbs and stores nutrients, then makes them available to the cells of the body. Exchanges respiratory gases with the environment, transports respiratory gases, nutrients, hormones, and heat. Around the body, brings extracellular fluids back into the circulatory system, helps the immune system fight invading organisms. Fights invading organisms and infections. Protects the body from invading organisms and harsh physical conditions, helps regulate body temperature. Regulates the composition of the extracellular fluids, excretes waste products. Institute for Advanced Studies, Job 15. Bacteria and archaea, the prokaryotic domains. The organisms most commonly found in such extremely acidic environments belong to the two major groups of prokaryotes, bacteria and archaea. The bacteria live in almost every environment on Earth. The archaea are a superficially similar group of microscopic, unicellular prokaryotes. However, both the biochemistry and the genetics of bacteria differ in numerous ways from those of archaea. Not until the 1970s did biologists discover how radically different bacteria and archaea really are. And only with the sequencing of an archaeal genome in 1996 did we realize just how extensively archaea differ from both bacteria and eukaryotes. Many biologists acknowledge the antiquity of these clades and the importance of their differences by recognizing three domains of living things: bacteria, archaea, and eukarya. The domain bacteria comprises the true bacteria. The domain archaea, Greek archaios, ancient, comprises other prokaryotes once called inaccurately ancient bacteria. The domain eukarya includes all other living things on Earth. Dividing the living world in this way, with two prokaryotic domains and a single domain for all the eukaryotes, fits with the current trend toward reflecting evolutionary relationships in classification systems. In the 8 chapters of Part 5, we celebrate and describe the diversity of the living world, the products of evolution. This chapter focuses on the two prokaryotic domains. Why three domains? What does it mean to be different? You and the person nearest you look very different. Certainly you appear more different than the two cells. But the two of you are members of the same species, while these two tiny organisms that look so much alike actually are classified in entirely separate domains. Still, all three of you in the domain eukarya and those two prokaryotes in the domains bacteria and archaea have a lot in common. Members of all three domains. E conduct glycolysis. E replicate their DNA semi-conservatively. E have DNA that encodes polypeptides. E produce these polypeptides by transcription and translation and use the same genetic code. E have plasma membranes and ribosomes in abundance. There are also major differences among the domains. Members of the eukarya have cells with nuclei, membrane-enclosed organelles, and a cytoskeleton, structures that no prokaryote has. And a glance at table 1 will show you that there are also major differences, most of which cannot be seen even under the microscope, between the two prokaryotic domains. In some ways the archaea are more like us, in other ways they are more like bacteria. Genetic studies have led many biologists to conclude that all three domains had a single common ancestor, and that the present-day archaea share a more recent common ancestor with eukaryotes than they do with bacteria. Figure 1. Because of the ancient time at which these three clades diverged, the major differences among the three kinds of organisms, and especially the likelihood that the archaea are more closely related to the eukaryotes than are either of those groups to the bacteria, many biologists agree that it makes sense to treat these three groups as domains, a higher taxonomic category than kingdoms. To treat all the prokaryotes as a single kingdom within a 5-kingdom classification of organisms would result in a kingdom that is paraphyletic. That one is, a single kingdom, prokaryotes, would not include all the descendants of their common ancestor. One of the three domains of life on Earth. Domain. Characteristic bacteria archaea eukarya membrane enclosed nucleus absent present membrane enclosed organelles absent peptidoglycan in cell wall present absent membrane lipid ester linked ether linked ester linked. Unbranched branch tilde unbranched ribosomes. 708,708,808 initiator turn of formal methionine. The common ancestor of all three domains was prokaryotic. Its genetic material was DNA. Its machinery for transcription and translation produced RNAs and proteins, respectively. It probably had a circular chromosome, and many of its structural genes were grouped into operons. Archaea and eukarya are more closely related to each other than they are to bacteria. Very ancient prokaryotes. LL bacteria. Origin R_E of life Bay archaea. 9. Eukarya. LJ ancient present time. Today's organisms all share this common ancestor. One of three domains of the living world. Many biologists believe that the three domains share a common prokaryotic ancestor. The relationships shown here, however, remain controversial. The archaea, bacteria, and eukarya of today are all products of billions of years of natural selection and genetic drift, and they are all well adapted to present day environments. None are primitive. The common ancestor of the archaea and eukarya probably lived. 16 Institute for Advanced Studies, Jodhpur. More than 2 billion years ago, and the common ancestor of the archaea, the eukarya, and the bacteria probably lived more than 3 billion years ago. The earliest prokaryotic fossils date back at least 3.5 billion years, and these ancient fossils indicate that there was considerable diversity among the prokaryotes even during the earliest days of life. The prokaryotes were alone on Earth for a very long time, adapting to new environments and to changes in existing environments. They have survived to this day, and in massive numbers. General biology of the prokaryotes. There are many prokaryotes around us, everywhere. Although most are so small that we cannot see them with the naked eye, the prokaryotes are the most successful of all creatures on Earth, if success is measured by numbers of individuals. The bacteria in one person's intestinal tract, for example, outnumber all the humans who have ever lived, and even the total number of human cells in their host body. Some of these bacteria form a thick lining along the intestinal wall. Bacteria in Antarctica and the oceans number more than 3 by 10 degrees registered. This stunning number is perhaps 100 million times as great as the number of stars in the visible universe. Although small, prokaryotes play many critical roles in the biosphere, interacting in one way or another with every other living thing. In this section, we'll see that some prokaryotes perform key steps in the cycling of nitrogen, sulfur, and carbon. Other prokaryotes trap energy from the sun or from inorganic chemical sources, and some help animals digest their food. The members of the two prokaryotic domains outdo all other groups in metabolic diversity. Eukaryotes, in contrast, are much more diverse in size and shape, but their metabolism is much less diverse. In fact, much of the energy metabolism of eukaryotes is carried out in organelles, mitochondria, and chloroplasts that are descended from bacteria. Prokaryotes are found in every conceivable habitat on the planet, from the coldest to the hottest, from the most acidic to the most alkaline, and to the saltiest. Some live where oxygen is abundant and others where there is no oxygen at all. They have established themselves at the bottom of the seas, in rocks more than 2 kilometers into Earth's solid crust, and inside other organisms, large and small. Their effects on our environment are diverse and profound. What do these tiny but widespread organisms look like? B. Escherichia, Leptospira, enterorgans. Prokaryotes and their associations take a few characteristic forms. 3 shapes are particularly common among the prokaryotes: spheres, rods, and curved or spiral forms. Figure. A spherical prokaryote is called a coccus, plural, cocci. Cocci may live singly or may associate in 2 or 3-dimensional arrays as chains, plates, blocks, or clusters of cells. A rod-shaped prokaryote is called a bacillus, plural, bacilli. Spiral forms are the third main prokaryotic shape. Bacilli and spiral forms may be single or may form chains. Prokaryotes are almost all unicellular, although some multicellular ones are known. Associations such as chains do not signify multicellularity because each cell is fully viable and independent. These associations arise as cells adhere to one another after reproducing by fission. Associations in the form of chains are called filaments. Some filaments become enclosed within delicate tubular sheaths. Prokaryotes lack nuclei, organelles, and a cytoskeleton. The basic unit of archaea in bacteria 1 is the prokaryotic cell, which contains a full complement of genetic and protein synthesizing systems, including DNA, RNA, and all the enzymes needed to transcribe and translate the genetic information into proteins. The prokaryotic cell also contains at least one system for generating the ATP it needs. In what follows, bear in mind that most of what we know about the structure of prokaryotes comes from studies of bacteria. We still know relatively little about the diversity of archaea, although the pace of research on archaea is accelerating. The prokaryotic cell differs from the eukaryotic cell in three important ways. First, the organization and replication of the genetic material differs. The DNA of the prokaryotic cell is not organized within a membrane-enclosed nucleus. DNA molecules in prokaryotes, both bacteria and archaea, are usually circular. In the best studied prokaryotes, there is a single chromosome, but there are often plasmids as well. Second, prokaryotes have none of the membrane-enclosed cytoplasmic organelles that modern eukaryotes have, mitochondria, Golgi apparatus, and others. However, the cytoplasm of a prokaryotic cell may contain a variety of infoldings of the plasma membrane and photosynthetic membrane systems not found in eukaryotes. Third, prokaryotic cells lack a cytoskeleton, and, without the cytoskeletal proteins, they lack mitosis. Prokaryotic cells divide by their own elaborate method, fission, after replicating their DNA. Prokaryotes have distinctive modes of locomotion. Although many prokaryotes cannot move, others are mobile. These organisms move by one of several means. Some spiral bacteria, called spirochetes, use a corkscrew-like motion made possible by modified flagella, called axial filaments, running along the axis of the cell beneath the outer membrane. Many cyanobacteria and a few other bacteria use various poorly understood gliding mechanisms, including rolling. Various aquatic prokaryotes, including some cyanobacteria, can move slowly up and down in the water by adjusting the amount of gas in gas vesicles. By far the most common type of locomotion in prokaryotes, however, is it driven by flagella. Institute for Advanced Studies, Job 17. Bacterial flagella are slender filaments that extend singly or in tufts from one or both ends of the cell or are randomly distributed all around it. Figure L1 flagella #075 pm. A bacterial flagellum consists of a single fibril made of the protein flagellin, projecting from the cell surface, plus a hook and basal body responsible for motion. In contrast, the flagellum of eukaryotes is enclosed by the plasma membrane and usually contains a circle of 9 pairs of microtubules surrounding two central microtubules, all containing the protein tubulin, along with many other associated proteins. The prokaryotic flagellum rotates about its base, rather than being as a eukaryotic flagellum or psyllium does. Prokaryotes have distinctive cell walls. Most prokaryotes have a thick and relatively stiff cell wall. This wall is quite different from the cell walls of plants and algae, which contain cellulose and other polysaccharides, and from those of fungi, which contain chitin. Almost all bacteria have cell walls containing peptidoglycan, a polymer of amino sugars. Archaeal cell walls are of different types, but most contain significant amounts of protein. One group of